We've all heard the promise of the ultimate cancer-fighting smart bomb. The idea that we can engineer a microscopic Trojan horse, designed to seek out and destroy cancer cells while leaving your healthy tissue completely untouched.
Right. It sounds like the absolute holy grail of precision medicine.
Exactly. But today we are looking at the reality behind these drugs, antibody-drug conjugates or ADCs, because if they are so incredibly smart, why do they still sometimes fail?
Yeah, it is a really profound paradox. I mean, the fundamental concept of an ADC is incredibly elegant, but the biological reality of the human body and of cancer itself is just, well, it's chaotic.
And that's exactly our mission for this deep dive today. We are unpacking a massive, comprehensive clinical review that maps out the lessons learned from setbacks in ADC development.
Yeah, we're basically going to explore the microscopic battlefield. We want to find out exactly how tumors fight back, look at the hidden toxicities, and unpack the futuristic AI-driven solutions being built to fix these issues.
So, to visualize this for you listening, let's toss out the boring textbook diagrams. Imagine a highly orchestrated bank heist. You have the crew, which is the drug. You have the vault door you need to crack, representing the target on the cell.
Right, and then you have the layout of the bank itself, which is the tumor's biology.
Yeah. And finally, you have the security system and the guards, representing the surrounding tumor microenvironment. If any one of those four elements goes wrong,
the heist fails. Exactly.
So let's start with the crew, the ADC construct itself.
So this isn't a single chemical compound. It's a complex three-part machine. First, you have the antibody, right? That's the navigator.
Okay.
It's an engineered protein designed to lock onto a specific marker on the cancer cell. Second, you have the payload, which is the ultra-toxic chemotherapy warhead.
The bomb.
Exactly, the bomb. But the critical point of failure is actually often the third component, the linker.
Right, the linker. That's the physical trigger connecting the navigator to the bomb, right?
Yeah, but it's far more than just a physical chain. The chemistry of the linker is arguably like the hardest part to engineer.
Wait, really? Why is that?
Well, it has to remain perfectly stable while floating in the neutral pH of your bloodstream, in enduring the turbulent journey through the heart and liver.
Right.
But the millisecond it gets inside the acidic environment of a tumor cell, that linker has to instantly degrade and release the toxin.
Oh, wow. So it has to be tough and fragile at the exact same time.
Exactly. If it breaks too early, you get systemic poisoning. If it doesn't break at all, well, you've just delivered a dud.
And there's another detail on that antibody crew member I found totally fascinating. The FAC region, like the tail end of the antibody. I always just assumed it was structural.
Most people do, yeah. But drug designers are actually tweaking it to talk to the immune system. The FAC region dictates how the body's natural immune cells react to the presence of the drug.
That is so smart. How they tweak it.
Well, sometimes engineers design this region to act as a distress flare. It triggers what we call ADCC. That stands for antibody-dependent cell-mediated cytotoxicity.
Okay, that's a mouthful. What does it actually do?
So when the drug binds to the tumor, that tail end basically signals nearby natural killer cells to swarm and attack the cancer.
It's calling for backup.
Yes. But interestingly, for other ADCs, designers intentionally strip that function away. They mutate the FCC region so the immune system ignores it entirely.
Oh, turning the drug into like a stealth delivery vehicle?
Precisely. You don't always want the immune system reacting too early.
Okay, so that's our crew. Let's talk about the vault door, the target on the cell.
Yeah.
Because it's not just a simple lock and key situation, right? The cell actually has to swallow the drug.
Yeah, this is a major stumbling block called internalization. The target antigen on the surface of the cell has to bind to the ADC, and then the cell membrane physically folds inward.
So it like swallows it whole.
Yeah, it pinches off to form a little bubble inside the cell. It's a process called endocytosis. If the target protein doesn't initiate that swallowing mechanism, the drug just stays stuck on the outside of the cell and does literally nothing.
That is wild. And to make it harder, the vaults keep changing. The review talks about intratumor and intertumor heterogeneity.
Heterogeneity is honestly the nightmare of oncology. Let's break it down. Intratumor heterogeneity means if you look at a single tumor mass, the cancer cells on the left side might be absolutely covered in these target proteins.
But the right side.
The literal neighbors on the right side of the exact same mass might not express the target at all. So the drug wipes out half the tumor and leaves the rest to grow.
And intertumor.
Intertumor heterogeneity means a cancer that started in the breast, for example, and spread to the liver, Well, it might have completely different targets in the liver than it does in the lungs.
So the key works on the first floor, but not the second floor of the bank.
That's a great way to put it.
Which brings us to the third part of the heist. The layout of the bank itself. The tumor type. Because even if the cell swallows the drug, the biological wiring dictates what happens next.
Right. The review mentions histology, oncogenic pathways, and ADC transporters. Basically, the payload's success depends on the cell's internal machinery.
So what do those actually mean in practice?
Well, histology refers to the physical structure and origin of the tissue. Oncogenic pathways are the specific mutated circuits the cell uses to divide and survive.
Okay, so if your payload is designed to damage DNA.
But that specific tumor type has a highly active mutated DNA repair pathway. The cell will literally fix the damage as fast as the payload inflicts it.
Oh man, it's like patching a hole in a boat while someone is still drilling it.
Exactly.
Oh.
And furthermore, certain cells express specialized transporters, which are proteins that shuttle molecules around. If the cell lacks the right transporter to move the payload to the nucleus, the drug is useless.
And if that isn't enough, we have the security guards. The tumor microenvironment, or the TME.
Yeah, the TME is arguably the most formidable physical barrier. This isn't even the cancer itself. It's the dense, poisonous jungle surrounding the tumor.
So how does the cancer build this jungle?
Solid tumors recruit surrounding healthy tissue and twist it to their advantage. They create high stroma density, which is basically thick, fibrotic scar tissue acting as a physical wall.
Right, like a fortress.
And the blood vessels inside tumors are incredibly leaky and poorly formed. That results in terrible fluid pressure that literally pushes drugs away from the tumor.
And then there's the extracellular matrix, right? The gel between the cells. The review says it's packed with extracellular proteases.
Yes. Proteases are enzymes. You can think of them as essentially chemical scissors.
Just floating around in the jungle.
Exactly. And if they bump into our ADC, they can prematurely snip the linker. That drops the toxic payload into the surrounding healthy tissue before it ever reaches the cancer cell's vault door.
Okay, let's do a quick mini recap here to make sure we're tracking this whole journey. We've got this highly engineered crew, the ADC construct with its antibody, the finicky linker, and the toxic payload.
Right.
But to pull off the heist, they have to survive the chemical scissors in the jungle-like microenvironment. Then find a cell that actually has the target, force the cell to swallow them through internalization, and hope the cell's internal layout doesn't just repair the damage instantly.
Yeah, that is an incredibly narrow path to success.
Yeah.
All four pillars have to align perfectly.
So here's a question for you, the listener, to chew on before we move on. If you were designing the next billion-dollar cancer drug, would you focus your research on sharpening the machete to cut through that physical jungle of the microenvironment, or would you focus on building a more complex key to trick the tumor's vault door?
It's a tough call, and the field is heavily split on that exact question. But the strategy you choose dictates the diagnostic tools you need, which brings us to a critical section of this review.
Right, the 7 key gaps to realizing true precision medicine. I have to play devil's advocate here. Right now, to see if a patient should get an ADC, doctors usually take a tissue biopsy and run a test called immunohistochemistry, IHC.
That's the standard, yes.
They stain the tissue on a slide to see if the target protein is there. But based on the chaotic, changing environment we just described, isn't relying on a single IHC test, like trying to understand the plot of a whole 2-hour movie by looking at one blurry photograph.
It is a severe limitation.
Yeah.
And it's precisely what gaps 1 and 2 in the review address. IHC is non-quantitative. It tells you if a target is present, but not necessarily how much of it is there.
And it misses the heterogeneity we talked about.
Completely misses the spatial and temporal heterogeneity. That biopsy tells you the target was present on a Tuesday in 1 millimeter of tissue. It tells you nothing about the tumors in the rest of the body or how they might change by Friday.
So how do we fix the blurry photograph problem? We can't just keep cutting people open.
No, we have to move toward dynamic, full-body mapping. Immunoheat imaging is a prime example. Instead of a surgical biopsy, we attach a safe radioactive tracer to the antibody and inject it.
Oh, and then you can literally track it on a screen.
Exactly. We put them in a PET scanner and watch, in 3 dimensions, exactly where the drug travels and binds across the entire body. But even that isn't enough, according to GAP 2.
Why not? If you can see it binding, isn't that proof it works?
Not necessarily. We need assays to measure the internalized payload kinetics, meaning we need to know what happens after the cell swallows it.
Ah, right. Just because it attaches to the surface doesn't mean the cell actually digests it properly.
Exactly. We need tools that track intracellular trafficking to confirm the warhead is actually detonating inside the cell.
Which leads us right into gap 3, resistance mechanisms. Let's say the drug gets inside and it detonates. The tumor doesn't just give up, does it?
No, it's a living, evolving organism. The mechanisms they use to become immune to these smart bombs are remarkably sophisticated.
Tell me about it. Like, what's the first line of defense?
Often, the tumor simply downregulates the targets. It just stops manufacturing the surface protein the ADC is looking for, which renders the drug completely blind.
That's frustrating. But what if the drug does get swallowed?
Then the tumor can actually alter its own digestive system. Think about the lysosomes. That's basically the stomach of the cell.
Right, where the ADC is supposed to be broken down by acid and enzymes so the payload can be released.
Yes. Tumors can physically alter the pH levels inside their lysosomes or reduce the production of specific proteolytic enzymes.
They just turn off the acid.
Basically. There is a well-documented resistance mechanism against a breast cancer ADC called TDM1 where this exact thing happens. The cell swallows the drug, but because the lysosome lacks the right chemical scissors, the drug just sits trapped in that cellular stomach forever, completely inert.
Wow. It neutralizes the bomb by just refusing to open the package. What else do they do?
They can mutate the payload's target. For example, some payloads are designed to bind to and destroy an enzyme called TOP1, which the cell needs to copy its DNA.
So the tumor changes the shape of TOP1.
Exactly. The tumor will mutate the physical shape of its TOP1 enzymes. The payload still enters the cell, but it can no longer lock onto the enzyme.
That's devastating clinically, isn't it?
It is because it causes cross-resistance. Suddenly, a whole class of drugs just stops working. And then, well, there's the most direct defense, efflux pumps.
I love this one. The cellular bouncers.
It's a very accurate way to visualize it. The cancer cell upregulates certain genes to build microscopic ATP-powered pumps right on its membrane.
And they just throw the drug out.
Literally. As soon as the chemotherapy payload is released inside the cell, these pumps grab the toxic molecules and actively spit them right back out into the bloodstream against the concentration gradient.
This is just wild to me. If tumors are this adaptable, if they can just build pumps to spit the drug back out, are we just wasting billions of dollars playing whack-a-mole? How do we ever get ahead of this?
Well, the authors of the review argue that the only way to get ahead is to outcompute the tumor. That brings us to gaps 4 through 7, which deal with biomarkers, patient selection, and drug design.
Outcompute the tumor? What does that look like?
First, to anticipate resistance, we need longitudinal liquid biopsies. That means drawing blood repeatedly to catch tiny fragments of shedding tumor DNA. It allows us to watch the mutations happen in real time.
Okay, catching it early.
But ultimately, we have to move away from human intuition and rely on machine learning to generate what they call composite predictive signatures.
Unpack that phrase for us. Composite predictive signatures. What goes into that?
It means integrating massive data sets that no single human brain could process. You take the patient's clinical history and that 3D imaging we talked about.
Okay.
Then you add multiomics. Genomics gives you the blueprint of the tumor's DNA mutations. Proteomics maps the actual physical structures of the proteins the tumor is producing.
And you just feed all of this into an AI.
All of this, plus the density of that TME jungle, goes into a deep learning AI. The model synthesizes these multidimensional features and outputs a composite signature. It tells the doctor, this specific patient, right now, requires this exact engineered ADC.
Let's pause and do another mini recap, connecting the dots on this diagnostic shift. We're moving from a static 1D view of cancer-like staining a single tissue slide to a dynamic AI-driven model.
Exactly.
Because we now know cancer fights back from the inside out. It neuters the drug in its lysosomes, it mutates its internal targets, and it literally pumps the poison back out. So the only way to counter that level of evolution is by feeding a patient's entire genetic, protein, and spatial profile into a machine learning algorithm.
To predict the tumor's next move. Yes.
So listener, a question on trust for you. How comfortable are you with a machine learning algorithm deciding which highly toxic cancer drug you should receive, based on a mathematical composite signature, rather than relying solely on your oncologist's intuition?
That trust becomes even more critical when we look at what happens when these predictive models get it wrong. Because despite all this targeted engineering, the toxicities, the side effects, can be devastating.
Yeah. Section 3 of the review is pretty heavy. It dives into the dark side of ADCs, and I really need you to clarify this for me. If the entire selling point of an ADC is that it's a smart bomb, only unlocking when it finds cancer, how are healthy cells getting caught in the crossfire?
Because biology is full of loopholes. Healthy cells sometimes inadvertently mimic cancer's vulnerabilities. The review highlights a fascinating mechanism called target-independent uptake, specifically through a process known as macropinocytosis.
Macropinocytosis, which means like large cell drinking, right?
Literally, yes.
Yeah.
Certain healthy cells in your body are highly active and naturally thirsty. Megakaryocytes are a great example.
What do they do?
They are giant cells in your bone marrow responsible for manufacturing blood platelets. To do their job, they constantly gulp down large volumes of surrounding fluid.
Oh, no. So if the ADC is in the fluid.
Exactly. If there are ADCs circulating in the bloodstream, the megakaryocytes accidentally drink them in. Once inside, the toxic payload releases, inhibiting the cell's ability to mature.
And that causes platelet drops?
Yes. The result is severe thrombocytopenia, a dangerous drop in the patient's blood platelets. The drug didn't misfire. The healthy cell just drank the poisoned water.
It's completely mechanical. It has nothing to do with the target. But what about the lung toxicities? The paper issues this massive warning about interstitial pneumonitis linked to a specific ADC called trastuzumab deruxtecan, TDXD. Why the lungs?
It comes down to cellular garbage collectors. Inside the tiny air sacs of our lungs, we have immune cells called alveolar macrophages.
Okay, so they clean the lungs.
Right. Their entire evolutionary purpose is to patrol the lungs, recognize foreign debris, and swallow it to keep the airways clear. Unfortunately, these macrophages frequently mistake the circulating TDXD for cellular debris.
So they swallow the bomb on purpose, thinking it's trash.
Yes. They swallow the drug, the payload detonates inside them, and the resulting mass death of macrophages triggers a cascade of severe, sometimes fatal, lung inflammation.
That is tragic. And beyond mechanical accidents, there's the patient's own genetics, right? Pharmacogenomics. Because two patients can receive the exact same dose of the exact same ADC, and one has catastrophic side effects while the other is totally fine.
Yeah, that often comes down to liver enzymes. The review points to a specific genetic polymorphism, which is a variation in the DNA called UGT1A128.
Catchy name. What does it do?
Well, if a patient inherits this variation, their liver is highly inefficient at processing and clearing out certain chemotherapy payloads, like irinotecan. The payload lingers and builds up to toxic levels in the blood.
Wow. Are there other payload-specific issues?
Definitely. We also see chronic issues with microtubule inhibitor payloads. These specific toxins damage the structural integrity of nerve endings. That causes chemotherapy-induced peripheral neuropathy.
Which is nerve pain, right?
Yes. Patients experience constant agonizing tingling or numbness in their hands and feet that can literally last for years.
This collateral damage is forcing a massive strategic pivot in how we use ADCs. The review outlines a path forward in Figure 3, suggesting we need to move these drugs away from late-stage metastatic patients and introduce them much earlier in the disease process.
Yes, that's a big shift in the field right now.
But if they are this toxic, why give them earlier? That seems counterintuitive.
It's all about evolutionary pressure. Think about it. Late-stage tumors have usually survived multiple rounds of brutal chemotherapy. They are hardened veterans.
Right, they've already built the efflux pumps and mutated their target.
Exactly. Early-stage tumors, on the other hand, are naive. Their locks are easier to pick. By hitting them early, we can exploit their molecular vulnerabilities before they have time to adapt. Furthermore, early tumors have more intact blood vessels, meaning the drug can penetrate the tissue more effectively.
But the trade-off is terrifying. You are giving a highly toxic drug that might cause permanent neuropathy to an early-stage patient who might have already been completely cured by their initial surgery. How do doctors balance that risk?
It's a huge challenge. The authors propose dynamically guiding the treatment using ultrasensitive blood tests looking for MRD, minimal residual disease.
How does that work in practice?
After surgery, you test the blood for microscopic traces of tumor DNA. If the MRD is 0, you hold back the toxic ADC. You don't risk it.
As if it rises.
If the MRD levels start to rise, indicating the cancer is silently growing, you hit it with the ADC in adaptive trials. It's essentially a real-time risk assessment algorithm.
Let's do our third mini recap here. We've discovered that the friendly fire from these smart bombs isn't random. It's driven by thirsty bone marrow cells accidentally drinking the drug and overzealous lung macrophages treating the drug like garbage to be disposed of.
Right.
On top of that, your liver genetics can dictate if the toxin builds up in your blood. To minimize this, oncology is shifting toward hitting naive early-stage tumors, using minimal residual disease blood tests as a speedometer to know when to accelerate treatment and when to hit the brakes.
That's a perfect summary.
So, listener, a tough question for you. If you took a genetic test and found out you had that UGT1A1 variation, making you highly susceptible to severe, painful toxicities, would you still consent to taking the most effective targeted cancer drug available?
It is the ultimate tightrope walk in modern medicine, balancing efficacy against the quality of life. And because of these massive hurdles, the resistance mechanisms and the toxicities drug engineers have had to return to the drawing board.
Which brings us to the final frontier covered in this review. The next generation of ADCs. If the tumor builds a taller wall, we just have to build a smarter Trojan horse. What do these next generation molecules actually look like?
They are turning into incredible multifunctional machines. A major breakthrough is the bispecific ADC.
Yeah, specific, meaning two targets.
Exactly. Remember how tumors can simply stop producing the target protein to hide from the drug?
Well,
a bispecific ADC has a heavily engineered antibody that looks for two completely different targets on the cancer cell simultaneously.
oh, that's clever.
The review highlights a construct called BLB01D1, which targets both the EGFR receptor and the HER3 receptor.
It grabs the tumor with both hands. Why is that so much more effective?
Because it cuts off the tumor's escape route. Historically, when doctors hit a tumor with a drug that only blocks EGFR, the tumor eventually adapts by activating a dormant HER3 signaling pathway to keep growing.
Oh, it's like a biological escape hatch.
Exactly. By targeting both receptors at the exact same time, the bispecific ADC slams the escape hatch shut while delivering the payload.
Okay, so if grabbing two targets is good, what about the warhead? The review mentions dual payload ADCs. Why send in a pigeon with one explosive when you can strap two totally different bombs to it?
Precisely the logic. Next-generation designs like dual payload TROP2 or CEACAM5 ADCs attach two entirely different classes of chemotherapy to the same antibody.
Give me an example of how that plays out inside the cell.
Let's say the payload carries a DNA damager and a microtubule inhibitor. If the cell uses its mutated enzymes to fix the DNA damage, the microtubule inhibitor still shreds the cell's internal skeleton.
Wow.
It mathematically bypasses cross-resistance because the tumor would have to simultaneously evolve two completely different defense mechanisms to survive.
And the most exciting innovation to me in this whole paper is rational combinations. This is where we stop treating the ADC like a lone wolf, right? We start pairing it with immunotherapy. The review specifically mentions combining ADCs with CD47 blockades and checkpoint inhibitors. Break that down for us.
This is where we see true biological synergy. So many tumor cells cover themselves in a protein called CD47. This protein essentially acts as a giant "don't eat me" sign, tricking the body's immune system into ignoring the cancer.
Okay, so they're hiding in plain sight.
Exactly. When we combine an ADC with a CD47 blockade, we are chemically ripping that sign down.
The cloaking device is disabled.
Yes. The ADC enters the tumor, explodes, and kills a wave of cancer cells. When those cells burst, they release a flood of tumor antigens, molecular debris, into the surrounding environment.
And then the immune system notices.
Because we've removed the "don't eat me" sign and added checkpoint inhibitors to take the brakes off the immune system, the body's dendritic cells recognize this debris. They sound the alarm, and the patient's own adaptive immune system floods the tumor microenvironment to finish the job the ADC started.
It's the ultimate 1-2 punch. The engineered drug breaks down the wall, and the patient's own immune system storms the castle. Let's recap this final leap in engineering. We are moving past single-target drugs. Bispecific ADCs grab two different receptors to cut off evolutionary escape routes.
All right.
Dual payload ADCs carry two different toxins to outsmart resistance. And by combining these drugs with immunotherapies, we use the drug's explosive destruction to actively wake up the body's natural defenses.
It's an incredible leap forward.
But this level of engineering leads to a massive question, listener. Can a single drug molecule become too complex? At what point does engineering a double-targeting, double-payload, immune-modulating molecule become literally impossible to manufacture reliably or affordably on a global scale?
That question cuts to the core of this entire review. If there is one overarching thesis to take away from all this data, it's that we must absolutely abandon the idea of ADCs as a simple lock and key mechanism.
Right.
They are dynamic, living interventions. They are constantly interacting with the patient's immune system, their liver genetics, the garbage-collecting macrophages in their lungs, and the tumor's constantly mutating defenses.
And to achieve the dream of precision medicine, our diagnostic maps, those machine learning multi-omics models we talked about, must become just as incredibly sophisticated as the drugs themselves.
A drug, no matter how perfectly engineered, is only as effective as the map guiding it to the target.
We've seen how antibody-drug conjugates are evolving from simple homing pigeons into complex AI-guided drone swarms carrying multiple payloads and directly communicating with our immune systems.
Yeah, it really is a fascinating time for oncology.
It really is. But here's something to mull over on your own as we wrap up. As we rely on these hyper-personalized machine learning algorithms and incredibly complex bispecific dual payload molecules, are we risking creating life-saving treatments that are simply too intricate and too staggeringly expensive to produce for the average patient? Is the ultimate setback of ADC development moving forward not biology, but accessibility? Thank you so much for taking this deep dive with us into the microscopic battlefield of precision oncology. See you next time.